Hello, can you hear me? Hello.
Yes,
Yeah. Good evening. So, in this week's problem solving session, we'll focus on the topics that were discussed in week 4 of NPTEL's genetic engineering course. To start with, the first question is the removal of terminal phosphate groups from DNA is done by, and four options are given. Option A, exonuclease, and followed by restriction endonucleases, alkaline phosphatase, and then DNA ligase. Whoever wants to uh, talk about an option can first, whoever knows the answer can first try justifying why other options are incorrect. We can wait for a couple of minutes and you can start answering. So if anyone would like to answer, you can raise your hand. Yeah, Vishwadip, you can start by telling us or justifying why an option is incorrect.
Sir,
I think there is an issue with your mic. Can you unmute and remute?
sir, now is it is clear.
Yeah,
So option B.
okay. What is that and what does it do?
So it cuts DNA from, inside.
Okay, so it removes, okay, it cuts DNA from inside. What do you mean by that? Can you rephrase your answer?
Uh, so um, so it um, cuts DNA from um, from between.
Okay, what other ways are there to cut a DNA?
So through using various enzymes like EcoR1
No,
or
no, no. What I'm
uh,
asking is, you
huh.
said this cuts DNA from between. Is that the only way to cut DNA?
No, another option is also there,
Hmm.
uh,
What is that?
like uh, uh, nucleases. I mean,
Yeah, you have the name right. It's you have one more option here, exonuclease. Does that?
exonuclease.
Yeah, what does it do?
It does cuts DNA from um, like from end
Hmm. What are the ends of a DNA?
uh, till I mean till 3 prime and 5 prime.
Hmm. Okay. Yeah, you're right. You have chosen that option A and B are incorrect. So, among nucleases, there are two types of nucleases. One category is endonuclease, those that can cut a covalently closed circular DNA. And the other that cannot do that but can cut from 5' end phosphate or the 3' OH. These are called exonucleases. So these do not remove the terminal phosphate groups. Rather, they chop down the DNA into nucleotides. So this is incorrect. Okay. Yeah, Chandrima.
Option D, DNA ligase.
What does it
It
do?
actually it makes the phosphodiester bond between the DNA.
Between what and what?
It joining the both DNA strands.
Between what and what?
Sorry, sir.
No. So you were saying it joins the DNA between two strands. Is that what you were saying?
Uh, there are two strands like in sideways. We have to ligate it. So that time we can use those DNA ligase to
Okay.
join.
Okay. But it does not um, remove phosphate, is it? It doesn't remove a phosphate group, is it?
No, it doesn't remove it.
Right. You're right about that. Except that. So now you need to be precise when you're talking about this. So the two strands of DNA are hydrogen bonded to each other. So ligation doesn't happen with among these two strands. Rather, it's just the same end. The 5' end of the same strand gets ligated to the 3' end of the same strand, or some other DNA's 3'. Okay, so yeah, Vishnudeep, you were raising your hand.
Sir, I mean, a DNA ligase, it joins uh, um, I mean, in a DNA replication, lagging strand, when lagging strand is formed,
Mhm.
then DNA ligase, it joins the that strand.
Can you rephrase
Uh,
it?
okay.
So during replication, you have leading strand and lagging strand.
So when and after I mean the then DNA ligase joins the Nix. I hope that is.
Okay, so you're trying to say in lagging strand, these are not formed together. You have something called as Okazaki fragments,
Yeah.
and that would mean you have one stretch of DNA and multiple small stretches that are complementary.
Yeah.
Okay, now among these, what is complementary? You have a 5' end and a 3' end, right?
Yes.
And you're saying this would, the DNA ligase would basically create a bond between these smaller fragments.
Yes.
Okay, it's partially right, partially incorrect. I so, but I would explain the mechanism with which that would happen. So based on this, we've eliminated all the other options and we go on with alkaline phosphatase. So we agree that this phosphatase is the uh, enzyme that would remove this terminal phosphate group. Now we'll look up its application where we'll also talk about how DNA ligase works. So normally when cloning is done, what we do is we choose two sites in a plasmid and we use restriction enzymes that would cleave. So your plasmid after digestion with two enzymes would look like this. It would have a 5' phosphate and a 3' OH and a complementary 5' phosphate and a 3' OH. Now once you get this, if you put a ligase, what would happen is this 5' phosphate and 3' OH would come close because they would come close to each other and a covalent bond will be formed. But if you put a phosphatase, what would happen is this 5' phosphate will be converted into 5' OH. This is the function of this phosphatase. Now, with respect to the answer that was said by Vishwaji, is it? Yeah. So what one should understand is this ligase has the ability to create a phosphoester bond between a 5' phosphate and a 3' OH, but it cannot repair nicks. Nicks are repaired by the DNA repair enzymes within the cell. So, Vishwadeep, like there is a difference between the answer that you told and how the mechanism actually happens. Is that clear to you?
Yes, sir.
So, unless and until you have, okay, are you aware of sticky ends?
Yes, sir.
Okay, so only if sticky ends are compatible, you can form this phosphodiester bond using a ligase. But
Yeah.
if nicks are there, these nicks have to be taken care of by the DNA repair system within the cell. These nicks cannot be repaired by uh, ligases specifically.
Okay.
Okay, cool.
Okay.
We'll go on to the next question. The next question is the eco R1 methylase adds a methyl group to the dash of the eco R1 restriction site. We'll take a couple of minutes, and whoever wants to answer can first talk about the eco R1 restriction site. Then we'll look at where it exactly methylates. Yeah, whoever knows can raise your hand. This session is supposed to be interactive, guys, so uh, you can try by answering whatever you know. So, what we would like to discuss here is about the Eco R1 restriction site. So, if anyone is aware of it, you can raise your hand. Yeah, Chandrama.
Sir, in EcoR1, the
Hmm,
restriction site actually it is in the sequence 5 prime
hmm,
GAA
hmm.
double T C 3 prime,
Right.
and it cuts after the
It cuts after the G. Okay.
G.
Okay. Right. Do you know about what a methylase is?
Yes, sir.
Do you know about a methylase?
Methylase is putting the methyl groups on the DNA strand to
Okay.
protect the DNA from cutting its own gene.
Hmm. Good. Yeah, Ananya.
The methyl group is added to the adenine side, so it does, as she said, it adds methyl group so that the endonuclease does not bind and cut the DNA.
Hmm. Right. Now, do you also know which A it is? Okay. It's okay. That would. Okay. Uh, Now, with regards to this methylation, do all enzymes have this ability to methylate their own DNA? Ananya,
It depends on what pattern the DNA has.
would it depend on the pattern or would it depend on the ability of the enzyme to cleave or the type of restriction modification system that's present? You can think about it. So there are different types of systems. Some are called restriction modification systems specifically because they not just have the ability to cleave the DNA, but they also have the ability to modify it. Like you said, it adds a methyl group to the A and it's at the second A at position 3 from 5' end, and this would prevent this enzyme from cleaving, which is why its own genomic DNA is not cleaved. Okay, now we'll talk about different types of restriction enzymes. So the question here is the cleavage site of type 1 restriction enzymes is dash base pairs away from the recognition site. So before anyone would like to answer, you can share what you know about the different types of restriction enzymes, type 1, type 2, type 3, based on which we'll further discuss and come to a conclusion. Um, if anyone would like to answer, you can uh, raise your hand. Ananya or Chandrima or even others could try attempting. Yeah, Chandrima. What are the types of restriction enzymes that you're aware of?
Actually, there are majorly three types of restriction enzyme.
Hmm.
One is type 1,
Hmm.
type 2, and type 3.
Hmm. Do you know why they're classified into such different types?
It is classified based on where they cut. Some of them cut at their restriction site. Sometimes they go away from it. Sometimes they go beyond that. So at where they are cutting, that is the main classification. On that, these three types are being classified.
Right. Yeah. So what you gave is a prompt description. So other than okay, we'll first look at the three different types. Did someone raise your hand? Did someone raise your hand? If yes, we can discuss. Okay, so Chandrima mentioned about how the three different uh, categories of restriction enzymes are based on the cleavage site. Like for example, type 1 cuts at around 1000 base pairs away from the recognition site. Type 2 cuts within its recognition site, which is why we use it for our cloning purposes. And type 3 cuts randomly some 20 base pairs away from the recognition site. But this alone is not a difference. Another difference that people have observed with respect to these different classes of restriction enzyme is that in type 1, The endonuclease and the methylase are on a single protein, whereas in type 2, these two are two different separate proteins. In type 3 also, these two are on the same protein. And a few examples of endonucleases are shown here. Now, based on this, the question that was asked is, the question that was asked is, for type 1 restriction enzymes, how many base pairs away is it from the recognition side? And that is 1000 base pairs. I hope this was clear. Yeah. The next question is, which of the following subunits of type 1 restriction enzymes provides specificity for the recognition site? They've given you four options: HSDR, HSDM, and HSDS, followed by MOD. Now, before anyone would like to answer, if you could try and abbreviate what the HSD, R, M, and S subunits stand for, that would be helpful in answering this question. If not, we'll discuss this. Yeah, Chandrama, you can talk about one option. It could be R, M, or S, followed by Vishwajit. Okay?
So, sir, any incorrect option?
Uh, no. Okay. You can either say what a particular subunit stands for. Like you have something called as HSD here, which is a
Yes, sir.
complex within which you have some subunits R, M, and S. Now, if we know what this R, M, and S would stand for, we'll be able to answer this question. So, if you know
Yes,
what the R, M, and S, we'll start with the
actually,
incorrect options itself, and then we'll narrow it down. After you,
uh,
Vishwajit will be going.
so I will be telling a wrong option.
Yeah.
So first option, that is SSDR. This
Huh.
is actually for the restriction of the enzyme,
So,
the
what does this
cutting,
do? Huh. And where does this
this
cut?
at the recognition site.
It's a type 1 enzyme. So, would it cut at the recognition site?
No, it will be going 1,000 base pair away from that recognition site.
Okay. Right. And we'll come to you next after Vishwajit. Yeah, Vishwajit.
Sir, wrong or right option?
You can talk about HSDM.
Sir, it modifies a sequence, I mean a DNA sequence.
So here, by modification, can we be more specific? What are the
Uh,
modifications that a DNA can undergo?
Sir, a foreign gene is added and it is been lighted that.
So you can take a look at this. So here we also talk about what is there in the complex. An endonuclease is there and a methylase is there. So what do you think is the possible modification that could happen? Yeah, Vishwajit.
No, I mean, I don't have any idea about it.
Okay, so here we were discussing about a particular modification that generally happens. So do you think that is the possible modification that you are referring to, methylation?
Huh, yes.
Right. Yeah. So methylation is the modification that we are talking about here. Right. Now, Chandrima. So what do you know about HSDS?
Actually, it is at that time recognizing the restriction sites, that portion where it is cutting.
Hmm.
Actually, at that time they are recognizing it.
So that is what recognizes the palindromic sequence.
Yes.
Right. Yeah. So like it was discussed, these have different subunits. Now this HSDM, it carries out the methylation. The HSDR is what will cut the DNA, and HSDS is what will identify the sequence, which is also called the restriction site. So these are the three different components in the complex. Huh. Now we'll talk about another particular enzyme. Here they are asking the restriction enzyme that cuts methylated DNA is. So they have given three options: EcoR1, HIN3, BAMH1, and DPN1. Now, in order to be able to answer this question, one can try to think about the different types of enzymes, classes, restriction enzyme classes, based on which we'll be able to answer. I'll discuss their relevance with respect to cloning in laboratory conditions. Whoever knows the answer can try to explain their rationale for the answer. Deblena, do you want to try? Aritra, Rajata, or Saurabhi. Chandraman and Vishwajit, you guys want to try?
So about the incorrect option.
You can directly go with the right answer.
Uh,
If you
that
know.
is that is DPN one
Okay, why did you choose this?
because the above three they all the you follow the restriction methylase system,
Hmm.
so they will not be cutting those methylated DNA.
Hmm. Whereas DPN1. So what does it say about the classification these three belong to?
Sir, that classification I don't know.
So these three would belong to type 2, right? In that case.
Yeah.
That is the kind of classification that I was asking for. Fine. And that way, DPN1 would not belong to type 2. Can we make that statement?
Yes, we can.
Right. Now, you chose this answer because you knew that these three options were incorrect and they belong to the same class. Is that why, or do you know something else about DPN1?
No, I don't know about DPN1.
Okay, good. Okay. Vishwajit,
Yes, sir.
yeah, do you know anything about DPN1? Or, uh,
No, sir. I mean, I just know that
hmm,
it it cuts DNA when it is methylated.
yeah, DPN1 cuts
Only
when,
that I know.
right, right. Okay, so now DPN1, this enzyme, unlike the other three enzymes, can cleave only when its recognition site is methylated. And we usually use this in laboratory conditions when we want to eliminate DNA or plasmid DNA that has been derived from cells after plasmid purification instead of making the plasmid through PCR. So that is a particular application of DPN1 which we regularly do in laboratory conditions to get rid of the bacterially derived plasmid. The next question, and this is a straightforward question. The BAM-H1 restriction site is, so this doesn't have anything to think. It's a straightforward question. It's GGATCC. We'll go to the next question. The molar ratio of vector to insert for ligation reaction is ideally option A 1 is to 1, option B 1 is to 10, option C 1 is to 3, and option D 1 is to 5. Right. Now, if anyone would like to go talk about the logic, you can raise your hand. You can take a couple of minutes. Okay. Yes. Uh, it's because it's just the four of us. We'll have it like a small discussion. What do you guys know about ligation? Anyone can unmute and talk.
You're joining a DNA.
Right. That is ligation.
I mean, a gene. Sorry.
No, DNA is more broad. It can be something that is not a gene as well. So that is fine. Now, what do you know about vectors and inserts?
Sir, the portion in which uh, um, any foreign gene is, uh, I mean, added.
That's called a vector, is it?
Yeah.
Okay. Um,
So the inserts are the DNA fragments that are introduced into a vector, particularly
okay.
containing a gene of interest.
Okay. Hmm. Now, what is so specific about the vector that we are introducing an insert into a vector and not putting the insert directly into a cell?
Because for a particular trait to be seen, we cannot give a direct to a cell. It will not work. Sometimes it can be treated as foreign and it will be degraded somehow by the cell.
Uh,
So we are using this way.
but plasmids are also, or vectors are also foreign to the cell, right? Need not be, need not be from the same cell, right? Yeah, someone had unmuted. So, Chandrima, you can think about this way. What you told about degradation is in a way right, but what would make some DNA less susceptible to degradation? Chandrima, can you hear me?
Yes, sir.
Okay, you have a cell and you have a DNA that you want to introduce within the cell. If you just put the DNA, you're saying it will get degraded for some reason, right?
Yes, sir.
Now, what other modification, if we do, can we ensure that the DNA that we are putting in is not getting degraded? Okay, I'll give you a hint. You can try to answer from that. Let's say we have a DNA, we are putting it into the cell. We are ensuring that the number of copies of this DNA is some thousand or so. Also, it is been modified according to the restriction modification system of the cell. Now, the chances that this DNA will be degraded will be reduced. Do you agree?
Yes, sir, I agree with it.
Huh. So, the reason why we need a vector is okay. There are two ways you can look at it. One is you make higher copies of the DNA for which you need an origin of replication. Your cell should ensure that whatever DNA you're putting in is getting replicated. Okay. Only if it is getting replicated, it will stay in higher copies. So even if some copies are getting degraded for whatever reason, others would still stay intact. That is one. The other is your vectors are what would have your selection markers like ampicillin or kanamycin, with which you will identify whether your vector and insert are getting selected. So whenever you grow cultures, you will grow with this antibiotic selection marker just to ensure that whatever you're growing has your plasmid with your right insert. Is that clear?
Yes, sir, I got the point actually.
Okay, fine. Now with respect to ligation, they've given us many options. So they say the ideal case. Now, what do you need to know about this? The first thing that you need to know is, do you guys know what molar ratio is? Does anyone here know what molar ratio is?
Actually, if we want to find a molar ratio of some composition of, and from that composition we are going to suppose we have to take a one compound, so we are according that the number of moles in that composition of that particular compound divided by total moles it has been given. So this is
That
molar.
is mole fraction, no?
Yeah.
Okay. I'll give you an example. You can think about this. Now, let's say there is a reaction. 2A plus B gives C. Now, when we talk about molar ratio, we talk about stoichiometry. What we try to say is, in order to form 1 mole of C, we need 2 moles of A and 1 moles of B. Okay. So, similarly, so what we, okay, so what we talk or what we're trying to say here is, we need 2 molecules of A that will react with 1 molecule of B to give 1 molecule of C. So, if that's the case, what we talk about molar ratio is how many molecules of a particular compound, which is vector here, and how many molecules of insert are needed. So these options could mean like 1 molecule of vector is to 1 molecule of insert. Is that clear? That's the molar ratio concept. Is that clear?
Yes,
Yes,
sir.
sir.
Okay. Now with respect to ligation, what we want to ensure is we want at least a few positive, true positive ligations. When we take 1 is to 1 of vector is to insert, what would happen is usually the size of your vector is larger than your insert. So the chances of collision to happen such that ligation would happen is less, because of which this 1 is to 1 ratio can be uh, disregarded. 1 is to 1 on the other hand is a lot. That would lead to too much of crowding effect, because of which you might not get any positive uh, clones. Now this 1 is to 3 and 1 is to 5, these are generally used based on the size of your insert. If your insert size is greater than 1 kb for a vector of 5 kb, then we use 1 is to 3. But if your insert is lesser than 1 kb, we use 1 is to 5, right, in practical conditions. But in theory, we use 1 is to 3. Uh, was that clear?
Yes, sir.
Yes,
Okay.
sir.
Okay. If you have any questions, you can ask me. I'll further try to clarify. Okay. The next question is, which of the following ORI sites allows the vector to replicate in both gram-negative and gram-positive bacteria? So, to think about this particular question, one can think about the different ORIs that you have, as of now, been studying. So, if anyone would like to answer, you can raise your hand. Do you guys know of any of these origin of replication sites? Rajata?
So just a minute, sir.
Yeah. Meanwhile, Chandrama and Vishwajit, even you can let me know what you know about any of these origins of replication, like call E1, F1. These might be familiar to you.
Sir, I'm not sure, but option B.
Okay, what do you know about that? Call E1?
No, I mean, I don't know, but I think option B is the correct option.
Okay, do you have a basis as to why you say that?
Because I have just, I mean, I just guess. Uh, I have
Okay,
no idea. Oh,
okay. Yeah, Chandrama.
Sir, about the option C, that is PSC 101, that was that first cloning vector which was in work the Herbert was working with. So that is why I know about this vector, and the F1 I have no information, but call even is a I only know on this PSC 101. but it doesn't grow in the both gram positive and gram negative.
Hmm. Okay, so you say PSC 101 doesn't grow in both gram positive and gram negative. So, okay, do you know in which set of organisms does it grow? E. coli does it? Okay, to think about this, Chandrima, you can think about from which organism this PSC 101 was isolated from, and what is the status with respect to gram staining of that particular organism, whether it was gram positive or gram negative. That will give you an answer. We'll listen to Rajata's answer and then we'll go ahead. Yeah, Rajata, you can unmute.
RP4 is from RP4 plasmid, which I guess is found in Pseudomonas species.
Okay.
And these are both gram positive and gram negative.
You're saying Pseudomonas is both gram positive and gram negative, is it? Or what
No,
is
no, no. I
conferring?
said RP4.
Why do you think it can uh, get both? Why can it grow in both gram positive and gram negative? Do you?
I'm not sure.
Okay, so whenever you look at these plasmids, you need to think about where this origin was initially found. Like for example, F1 is from F1 bacteriophage, if I'm not wrong. And this PSC 101 is from Salmonella, some species of Salmonella.
Salmonella.
Yeah, Salmonella. So Salmonella belongs to gram positive or gram negative, Chandrima?
Gram negative.
Gram negative, right. So that would mean possibly this can be used in E. coli, which is also gram negative, but not in some gram positive species. Col E1 is from, it's based on colicin. Colicin is a toxin that is produced by E. coli, again gram negative. Whereas this RP4, it it has been said that it can grow in both. So this is because the origin of this RP4 is that it was found in some conjugative transfer apparatus. So what do we mean by this is that if there is one cell, if some other plasmid is, if this RP4 based plasmid is entering as that cell, then it would allow for conjugation, meaning more transfer of plasmid and nutrients, which would in a way allow it to act on different variations across the cell wall, cell membrane, which is why this possibly could help in both, could work in both gram positive and gram negative. So if one wants to look at this, they can look at the RP4 conjugative transfer apparatus, and that would help us understand how this origin is specifically non-specific, can go through both gram positive and gram negative. And another way to look at this is, I was mentioning that F1 is a bacteriophage. Uh, but it could only infect stains of E. coli and not something that is gram positive. So if there is some other plasmid, some other phage that could infect both gram positive and gram negative bacteria, then That origin could also sustain in both gram-positive and gram-negative cells. So that is the logic behind this. The next question is, a covalently closed circular plasmid, when exposed to dash, transform into an open circular form. Four options are given here. You can try to answer this by starting off with any option that you think is incorrect. Anyone could unmute.
So the option A, DNA ligase, it can't be because it is not exposing the open circle. It is rather than joining that strands. That is why it can't
Okay,
be.
first we'll talk about what covalently closed circular plasmid is and open circular form. Do you know what covalently closed circular plasmid is, Chandrima?
Actually, in covalently closed circular plasmid, then that plasmid, they the nicks are being covalently linked. Actually,
How is it different from open circular form?
in open circular form, there are some nicks actually in the DNA. So that is why it is opened in that circular plasmid also open
Uh,
way, but in this case it's not.
okay. Do you know about one of the options that is shown here, DNA gyrase?
Actually, DNA gyrase is actually separating the strands of the DNA.
And
Actually,
okay,
that
it,
I know actually.
it would separate the strands. So what does it do? Does it denature? If it's separating the strand, does it denature?
No, it will be not.
So it is separating. What is happening after the separation? Does it also ensure that it is forming the original plasmid it was earlier?
Uh,
So think about this. You have, wait, let me take a thread. Uh, let me take a thread. Now consider, I hope you're able to see this. Consider this as your open circular form. One minute, I would unblur this. Okay, I hope you're able to see. Consider this as the open circular form and this as the closed circular form. In all cases, what you observe is the DNA is covalently closed. It is not covalently broken. So, what exactly is the difference between the open circular and the closed circular form? So, uh, Chandrima, like it was mentioned, I think I made a mistake. What I wanted to say is usually the DNA is compact like this. I hope you're able to see this. It's compact like this. Its covalent bonds stay the same. What DNA gyrase does is it removes helicity because this is a stretch of double-stranded DNA that are covalently closed, and because this is helical in nature, positive and negative supercoiling would happen. This DNA gyrase is involved in reducing the supercoiling. It wouldn't break it open. That is its function. And you were right about the difference between the closed circular and the open circular form. The open circular form has a nick, right? Uh, Now, what would create a nick within a DNA? Ha, Chandrama.
In anyhow that the phosphodiester bond between them has been broken down.
Ha.
That
So, what will break the phosphodiester bond? Would it be an exonuclease or an endonuclease?
it will be an endonuclease.
Right, right. So, that is what we wanted to discuss in this question. Yeah. So, the next question is the ampicillin resistance gene from PBR322 is derived from. So, this is there is not much to think about this question, so I'll directly go to the answer. It is derived from this RSF2124. So if we look about this PBR 322, the ampicillin resistance is from RSF 2124. Tetracycline resistance is from PSC 101. It has two selection markers, and the origin of replication is from PMB1. The next question is blue white screening cannot be performed with bacterial colonies transformed with recombinant. Option A PBR322, option B PUC19, option C PET28A, and option D PET26B. So anyone who would like to answer this question, anyone who would like to answer this question can start by explaining what blue-white screening is. Yeah, Chandrima.
Actually, blue-white staining is used to determine whether the plasmid DNA has been transformed or not, or the recombinant DNA is present in that cell, host cell in which we are injecting or not. So for that, in that vector, we are having the LAGZ gene, which
Hmm,
is coding for beta galactosidase, and we also use a substrate, colorless substrate called XGAL.
hmm,
So actually, when this beta galactosidase is in active state, it will give it will oxidize that XGAL to an indolite derivative, which is giving a bluish color. And if we break that LAGZ gene, then that gene is not being functional and that enzyme will not be producing. So if we are giving that substrate, there will be no color change. It is used to determine whether that recombinant DNA is present or not. Actually.
okay, if that is the case, what is the purpose of a selection marker?
So it is to check whether the DNA, that the foreign DNA which has been placed in that vector,
Hmm.
is it giving any different factor or any change in it or not, to make a create a difference between that.
Can you rephrase it? I didn't clearly get your answer. So every plasmid has selection marker. Like for example, in the previous case, we saw PBR 322 where it has both ampicillin and tetracycline. So after introducing this DNA within the cells, if we grow it on a tetracycline plate or an ampicillin plate, we will know that the cell has taken up this DNA, foreign DNA, right? So why would we need blue-white screening?
Actually, in what in the selection marker we are having many multiple cloning site. If we cut from that restriction enzyme,
Huh.
so that we are cutting the restriction enzyme, then that gene. So if that gene we are cutting, there will be that property of that gene will be not getting it at that time. So we can differentiate at on that basis by using that substrate, or whenever any phenotypic change or any kind of change we can see.
True. Okay. You are right about this part where you mentioned that we have these selection markers, but if the MCS or the restriction site is present within the selection marker, then the selection marker will not act. In those cases, we need to be able to differentiate whatever colonies we get based on whether the DNA is within the cell or not, which is why we use this method. Right. Now I'll go through the procedure again, like what you were mentioning. That is right. You have this LAC-Z, which codes for beta galactosidase, which converts the gal substrate to form a blue precipitate. All of this is right. Now there are now how do you decide whether a plasmid can function for this particular process? Like they have
We
given a
can
list of
see,
plasmids. Huh, yeah, we can.
we have to see on those plasmids that laxy gene is present or not. Exactly.
Right, that is the right answer. PUC19 has LAC-C gene. PBR 322, no, it's the other way around. PBR 322, it has the LAC-C gene, whereas PUC19 doesn't, or PET28A doesn't, or PET26B doesn't, which is why PBR 322 cannot be used for this particular process. Right. Okay. Next, we move on to alkaline lysis. So, what is the correct sequence of steps during plasmid isolation by alkaline lysis? So, now with respect to alkaline lysis, does anyone know why we do alkaline lysis? Yogita? Yeah, Chandrama.
Actually, in alkaline lysis, we are using so that we can degrade the cell membrane, which is quite is slightly acidic in nature.
The cell membrane is
That is why using.
negatively charged. The cell membrane. Okay. So, if it is negatively charged, why can't we use any other method? Like the cell membrane is made up of lipids, right? Now, if we put a detergent, that would lyse the cell. Anyways, it will break open the membranes, right, Chandrima?
Yes, it is possible.
So, why would you need alkaline lysis method then? Is it about the lysis of the cell, or is it something to do with differences of plasmid and the DNA in alkaline conditions?
Actually, what is where they work? They are denaturing the chromosomal DNA. As our gene of interest here is plasmid DNA,
Hmm,
hence we are denaturating using the alkaline lysis method,
hmm.
and we are taking that plasmid DNA.
So the question was why we do alkaline lysis, right? So you can think about it this way. You have a cell that can be broken apart by any method. You can use physical methods like sonication also to break open the cell. So it is not for breaking opening the cell. Rather, the plasma DNA is very small compared to your genomic DNA. It is about 1 by 500 or 1 by 1000 of the length of your genomic DNA. So when you denature, not just your genomic DNA denatures, your plasmid DNA also denatures. Your proteins also denature. Do you agree with that?
Yes,
Right. But when you're renaturing by precipitating, what would happen is your plasmid DNA, because it's a very small size, the individual single strands can form hydrogen bonds and it can form a double stranded plasmid. But your genomic DNA cannot do that. That is the basis of alkaline lysis method. Was that clear? Or I'll explain that again if that was not clear.
sir, this is not clear.
It's not clear. Okay. So what I'm trying to say is you can break open the cell by any kind of method at any pH. That wouldn't really affect your ability to break the cell. Even acidic pH would break the cell. It's not about the ability of the membrane. Was that clear? Do you agree that you can break open the cell by different methods and it is not that because the membrane is negatively charged that we use alkaline method? Do you agree with that?
Yes, I am agreeing.
Okay. Now you're breaking opening the cell. Once you break open the cell, what do? You denature to remove all the you want to separate your proteins from your nucleic acids because you want your plasmid DNA, right?
Yes, we are.
So first you add STS and other reagents that would denature your proteins. Then this is done. Now you have only your nucleic acid fraction. In this nucleic acid fraction, you have both your genomic DNA and your plasmid DNA. And with respect to size, if you consider, your genomic DNA is at least 1000 times larger than your plasmid DNA. Do you agree?
Yes, sir.
Right. Now, in this case, under the environmental conditions, it is very easier for the plasmid. The single strands of, okay, by denature, what we mean is we break the hydrogen bonds of the plasmid. We are not removing any covalent bonds. We are only removing the non-covalent interactions. So all the hydrogen bonds between the two strands of the plasmid is broken down. So you have two single strands of the plasmid. Similarly, you have two single strands of your genomic DNA also. But during this condition, it is very easier for a plasmid DNA, because it's of very small size, to come close together by chance and form all the hydrogen bonds one after the other. If one forms, it would come like this. Because these hydrogen bonds are forming, then this would come. That way, the entire plasmid, it's easier because it's relatively small size compared to your genomic DNA. But your genomic DNA cannot really renature that way. Is this point clear?
Yes, sir.
Now, because this is happening, you can precipitate it, you can take it away. So that is why you can specifically separate plasmid using alkaline lysis method. So it is the differential ability of your genomic DNA compared to your plasmid DNA, because of which your plasmid DNA can renature and can be precipitated. Is that's the alkaline lysis process? Okay.
Yes, sir.
Okay. Now, given this background, what do you think will be the sequence of steps during plasmid isolation by alkaline lysis?
This is option B.
Hmm. Right. So it starts with alkaline lysis, then renaturation, then deproteination, and finally alcohol precipitation. Right. Yeah. So here it's just a brief protocol. So we have NaOH and SDS. SDS solubilizes the membrane. NaOH breaks down the cell wall, and it disrupts the hydrogen bonding between the DNA bases that converts the double stranded DNA, including your genomic DNA and your plasmid DNA, to single stranded DNA. This is called the denaturation step. Now, SDS will also denature the proteins. After this, we add potassium acetate. This will decrease the alkalinity of the mixture. Now, under these conditions, the hydrogen bonding between the bases of the single-stranded DNA can be re-established. So the single-stranded DNA can renature to double-stranded DNA. Now, this is the selective part. So for plasmids, it's easy to renature, but for genomic DNA, it's impossible to anneal. So this is what we were discussing earlier. The next question is, cause sites are present in four options are given. Yeast episomal plasmid, yeast artificial. Option C is AC MNPV, and option D is bacteriophage lambda based vectors. You can take a couple of minutes and then raise your hand. We can start with discussing what cause sites are, then we'll go through the different options. Okay. Yeah, Rajita. You can start by describing what cost sites are.
So, cause sites are specific DNA sequences found in lambda and related phages
Okay.
that play a critical role in packaging and circularization of the phage genome. They
O-
help the phage to, they enable the phage to replicate inside the host.
What would so is that an origin of replication? You're saying it would help the phage to replicate within a host. That would mean it's the origin of replication, right?
No, sir.
No, you can uh, try explaining it in a different manner. Do you know the full form of corset? Does it have a full form? Yeah, Rajata. Do you
Well,
know?
I'm not sure, but is it
Huh.
cohesive sites?
Yeah. So corsets are the full form of corsets is cohesive sites. So what does it? So now you can try to think about why it would help in repackaging. That's what you were mentioning. Replication also would come, but first we'll talk about repackaging. Why do you think it will be helpful in repackaging? Okay, what are cohesive sites?
They're specific DNA sequences.
Specific to what?
Phages.
Okay, but okay, what else do you know about this?
So not much, sir, just shallow.
No issues. So cohesive means it's similar to sticky ends. If you can look at the image here, the ends have the ability to re-anneal. This is what they meant by repackaging, wherever you have learned this from. So whatever is linear can form a circular form. So now this is repackaging. Is that clear?
Yes, sir.
Okay. Now repackaging is done. After repackaging, it has to undergo replication. Now, if it is in the linear form, do you think replication is possible? Do you think replication is possible when the DNA is linear form?
It's difficult.
Which is why you need a circular form. So that is why this cause site, that is how this cause site is helpful in both repackaging and replication. Does that make this clear for you?
Yes, sir.
Okay, good. So yeah, so the answer is bacteriophage lambda-based vectors. These have these cos sites. The next question is which region of EMBL3 vector is replaced by the insert? Option A left arm, option B central stuffer, option C right arm, and option D cos sites. You can take a couple of minutes. Yeah, Tyasa. So we were looking at this question. Which region of the EMBL3 vector is replaced by the insert? Left arm, central stuffer, right arm, and cos sites. You can take a couple of minutes and you can try to answer this question. Hello, can you hear me? Yeah, if does anyone know what the EMBL3 vector looks like?
Sir, I don't know, but it is a bacteriophage lambda-based vector.
Right. Okay. So each of these have leading regions, right? Left and right, based on which their directionality is given for these lambda-based vectors. Are you aware of that, Chandrima?
No, sir, I'm not aware.
Okay.
No, sir.
The proteins that are being encoded by these vectors, they have a particular nomenclature, right? L region, R region in the vectors. Are you aware of that? Okay, no issues. So usually they are divided into different parts. One you have as a left arm, one you have as the right arm, between which there is something called as the central stuffer. Now, what one needs to know is that the restriction sites are present between the central stuffer. So if we are going to use these sites, it would mean that we will replace the central stuffer with our gene of interest. So to answer this question, this insert will replace the central stuffer. Uh, was that clear, Chandrima? I can add more references regarding the nomenclature, left leading, right leading part in phages. I'll add that to this presentation so you can look at it later. And the last question for today's class is the tetracycline resistance gene produces which of the following? Ribosomal protection proteins, beta lactamase, neomycin phosphotransferase 2, and chloramphenicol acetyltransferase. So, does to answer this question, yeah, you can unmute. To answer this question, you can first think about uh, where tetracycline acts. That will help us answer this question. Yeah, Chandrama, where do you think tetracycline acts?
Actually, tetracycline acts on the translation process. It stops the translation process in that
Okay,
bacteria.
okay. So it's when you say it stops translation, so you
Actually,
ah
in tetracycline, it works when means it works on that translation process and blocks that translation process. Tetracycline working.
okay. So among the options given here, so what do you think will be the right option if that's the case? Hello. So you said it works in translation inhibition, right? So if it is going to work in translation inhibition, then the resistance gene, where should it work?
Ribosome.
Yeah, you're right about this. So how it works is, like you mentioned, tetracycline sits on the A site and it prevents translation. So now the resistance gene would not allow tetracycline to set here, thereby protecting the ribosomal proteins and ensuring the translation happens. So this is how this process happens. And with this, we come to the end of this week's problem solving session. If you have further doubts, you can ask me, or we can end the call. Cool. There looks like there are no doubts. Thank you for attending. So next week also on Monday we'll have the class at the same time. Thank you.